lesson for paper 2. I am just going to kind of walk you through what we're going to do today as people join the live lesson, and I'm going to just explain to you a couple of things that I think is going to come up in this paper, in paper 2, because I know a lot of you are probably interested in what to expect in this upcoming paper tomorrow, and the likelihood of certain things being asked because of what was asked, or should I say not asked, in paper 1. Now, first of all, paper 1, I believe, was pretty simple. Many of you said the same thing. And so what does that mean for paper 2? Historically, paper 2 is always more difficult than paper 1. And the reason for that is not because the content is harder, but because it's harder to get full marks in paper 2 because there's a lot more explanations that are not anatomical. So that means you're not describing anatomy, you are describing speciation, bipedalism, you're describing genetics and how things are inherited. So technically speaking, paper 2 is generally more challenging because of what you're doing. Now, what should we expect tomorrow? Well, we didn't draw anything in paper 1 on Friday. So in all likelihood, you're going to have to draw something, right? And you're probably thinking it's going to be between a diagram or a graph. I don't think they'll make you do both, but they will definitely make you do one of those two things. And when it comes to drawings, the only thing you really draw in paper 2 is a phase of meiosis or a chromosomal arrangement, like what do the chromosomes look like at the end of meiosis. If they make you draw a graph, well, then it's going to be either a bar or a line. It might be a histogram, but they drew a histogram last year and in the supplementary paper, so I don't think it's going to be that again. It might be a line. And as always, just be prepared for a pie chart. I'm not saying it's going to be in the exam. We just need to be prepared for one to come up. Okay, now just to tell you what we're going to do is we're going to do a little warm-up activity just to get into the right mindset. And then obviously, as always, you can put your questions in the chat and I see them and I can go back to them and I can answer them. But just to get our minds warmed up and on the right track, we're going to do this past paper question, very simple, on DNA. This particular one I have chosen is from the supplementary exam from 2024. Why did I choose this one and why do I suggest you all go and try this entire paper today? Is because whoever set this exam set the exam you're writing tomorrow. So generally the person who sets the June supplementary exam is the same person as the final examiner. So they set 2 papers in a row. And so I suggest you go and practice this one. I got this one off testpapers.co.za. Unfortunately, there is no memo yet. So I know it's a bit frustrating to have a paper with no memo, but it's because the memo is not official yet, so they don't kind of put it out. However, I do know one place you can get the memo. I actually know where you can. You can go to, if you want the memo for this exam, you can go to the WCED website, which is the Western Cape Education Department website, and they actually have the memo under the folder where they keep all the matric exams. You'll find it where you scroll down and you'll say exam resources and past exam papers. You should be able to find the memo there as well. Okay, cool. So let's just do a quick little warm up. And then just remember, if you come in or out of the live lesson, this is saved and posted later on. Okay, let's do a quick little warm up. So it says the diagram below represents 2 nucleic acids, okay? And at first glance, I'm hoping that everybody can automatically identify which one is DNA and which one is RNA. So let me just make sure we all do know how to do that. So if we go a little bit closer. So on the left-hand side here, diagram 1, this is most definitely DNA. And how did I know that? Well, the most important thing is it's double-stranded. So we've got 2 strands on either side. And also we have some hydrogen bases in the middle, hydrogen bonds in the middle here. So we have our weak hydrogen bonds down the middle. And so those 2 things already tell me that we've got DNA. On the left-hand side here, on the right-hand side, we've got RNA. And I know that that is RNA because it is a single strand. As you can see here, there's no opposite complementary strand on the other side that mirrors it. And also you'll notice that all of my nitrogenous bases here don't have any bonds connecting to anything. Then what I also tell my students to do is that if there are any labels on the diagram, label them before you begin. So if we look at A is pointing to the bond here, which is a hydrogen bond. And B is pointing to a component of a nucleotide. So B is pointing to this round backbone structure here, which is the phosphate. So we've got the phosphate, the sugar, and then we've got the nitrogenous base. And just remember the differences between the names of the two sugars. I just want to remind you, because a lot of us make this very, very fatal mistake in the exam. This is RNA. Therefore, this is ribonucleic acid. Excuse me, not ribonucleic acid. I'm talking nonsense. This is ribose. Okay, that's ribose. This sugar here on DNA is deoxyribose. So a lot of us make this fatal error that we don't know the differences between the two sugars. So please, 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 please know that it's deoxyribose and ribose. Now, before I go into the actual questions below, I'm just going to have a quick look at the chat. People are just speaking about weak hydrogen bonds. Yeah, that's what I'm talking about over here. That's a hydrogen bond. You can write it out in full, weak hydrogen bond, but I'm just writing it in shorthand now because I just want to label it quickly so that we can come back to it. Okay, so let's move on to, let me go back. Okay, let's move down here. So it says identify, I don't know why all my labels have disappeared now. What's my issue? Oh, it's because the page is moving. There we go. Okay, so let's go down to our question. It says identify the nucleic acid shown in diagram 1. We've already done that. That's DNA. Then it says identify A and B. So as I said earlier, A is our weak hydrogen bonds, and then B is our ribose sugar. Okay. I mean, no, it's not our ribose sugar. What am I talking about, guys? What am I talking about? B is our phosphate. It's our phosphate. Sorry, guys. It's because I was so caught up in labeling our ribose over here. There, B, phosphate. Okay. Number 3 says, name the type of sugar found in the nucleic acid represented in diagram 2. Well, luckily for us, I already labeled that, which is going to be our ribose sugar. So that's our answer here now, ribose sugar. And then it says number 144, which diagram 1 or 2 represents a nucleic acid that is used for tracing human ancestry and paternity testing? Now, it's quite interesting because I'm sure that some of you are maybe not 100% sure which one would you use. So first of all, tracing human ancestry. The only information that we use to trace ancestry comes from mitochondrial DNA. And so when we are testing for that, we are not going to be looking for RNA, because if we think about it, if I say here mitochondrial DNA, the answer is in the fact that it is mtDNA, which is tracing human ancestry, which means that we're going to be using in our diagram here, diagram 1, right? But then for paternity testing, what do we use for paternity testing? Well, we also use DNA for paternity testing. So the answer is also number 1. I know that because there are 2 options here, you're probably thinking the examiner wants you to go 1 or 2 or 2 or 1. But just so you know, the examiner is allowed to do this, and it's allowed. You are allowed to use the same number twice, if that makes sense. Just because you're 1 and 1, your answer is 1 and 1, doesn't mean that you're wrong. Okay, you don't have to have two different answers. Okay, don't fall into that trap, please, everybody. Okay, let's move on. Now, that's some basics to DNA and genetics. Or not genetics yet, just DNA and the structure of the DNA. If there's any questions pertaining to DNA, I'd love for you to ask them now, because if you don't ask anything now about DNA, what I would like to do is do a little warm-up question on human evolution and this phylogenetic tree. So I'm just going to have a look. You guys can read the question in the meantime. I'm just going to have a look at the chat and see if there is any questions that you guys maybe have on DNA. Specifically the structure of DNA, how to tabulate it, all that kind of thing. Let's see. Okay, there's a question for what is RNA used for? So RNA, everybody remember, is used for protein synthesis only. And then somebody asked me to go through protein synthesis, as well as also somebody asked here, could I please give them tips for codons and anticodons? So I think let's see. I think everyone else here is saying protein synthesis, DNA replication, DNA mutations, blah, blah, blah. Okay, so let's do that quickly before we do this human evolution question, which we can do definitely. Let's just add a page so that I can do that. Okay, so let's just quickly go over protein synthesis, and I can do mutations and everything at the same time. Okay, so when we speak about protein synthesis, we are talking about two steps. We are talking about transcription, and we are talking about translation. Now, the difference is here. Transcription occurs inside the nucleus, whereas translation is in the cytoplasm, and also it occurs at the ribosome. Okay, so technically the ribosome in the cytoplasm, that's the difference in the locations. And then in terms of the processes, transcription is essentially copying a section of DNA into messenger RNA. And the opposite side for translation, what is the purpose, is the reading and the creation of proteins with mRNA and tRNA, which is, remember, transfer RNA. So if that's like this short little summary between the two, we've got their locations, and then we've got the differences in their process. Now, what does that visually look like? So you should all expect, you're all expected to know what it looks like, translation, and transcription. So this is the nucleus. And what you often have is like a piece of DNA that is wound up on the inside like that. And what they do is they take a section of it and they unwind it. So you end up getting like a section here that's open like that. And this section here, you will see there is, of course, your DNA triplet bases on both sides, keeping in mind that they will have complementary bases on the other side. So these are DNA base triplets, okay? And what we need to do is we are going to create a template, and we are going to have, let's call this side strand 1 and this side strand 2, okay? Now, what happens is along comes the free-floating nucleotides, which, remember, these are RNA nucleotides. They're floating in the nucleoplasm. You've got adenine floating around. You've got guanine, cytosine, and uracil. Remember, in mRNA, there is no thymine. So if we were to create a template of strand number 1, our mRNA, which I'm just going to draw on the side here so it doesn't get in the way, or actually, you know what, I will erase this just so it's not in our way. Our mRNA is going to form here. So because thymine doesn't exist in RNA, this is going to be U for uracil, another U for uracil because they're complementary, and then T still thinks it joins to A. And so we have now built our first little piece of mRNA. Okay, that piece of mRNA is going to detach and it's going to exit out of the nuclear pore into the cytoplasm. And then this piece of DNA rewinds, rezips, and it is now protected. It never leaves the nucleus. I think it's very important before I continue to say the following things. When you are learning for transcription and translation, you should be using your exam guideline description. You will find it in one of the first few pages describing translation and transcription. You need to learn that off by heart. Don't learn anything else. If you have my study guide, I literally use the exact explanation from the exam guideline. So if you use my study guide or you use the exam guideline, those are the only two things that I would use. I wouldn't use your textbook. I wouldn't use anything else. That's what you have to learn. Now, if this is transcription that's finished, we're now left with a piece of mRNA. And I'm going to add in a few more letters to work with because I want to show you how this all works. So here is my little piece of mRNA. And now let's just break down what you're looking at here. So the mRNA is now in the cytoplasm. And what you're looking at is sets of 3 letters called codons. And there is another codon here and another one here. So if anyone ever asks you in an exam how many codons are there here? 3. There are 3 codons here. If they ever ask you how many amino acids are they coded for, it's also 3, because for every 1 codon, it codes for 1 amino acid. Okay, so that's how it works. So there's 3 codons and therefore 3 amino acids. So now what happens is we take our piece of mRNA and it's going to go into our ribosome. And I'm just going to remove some of this so it's out of our way. And so our ribosome is this like round, kind of looks like a coffee bean shaped structure that floats in our cytoplasm. And it reads the 3 letters at a time. So it's going to read these first 3 letters, the codon first. And what's going to happen is floating in the cytoplasm, there are going to be tRNA molecules. And those tRNA molecules, if I draw one here, literally sitting at the base, have complementary letters that are going to link up with this. So if this is an A, that will be a U. If this is G, this will be a C. And if that is C, that will be a G, because they're complementary. Sitting at the back of the tRNA is an amino acid. And I'm just going to write A for amino acid. And so this is a tRNA or a transfer RNA molecule. It's going to come in and it's going to attach at the bottom here. So what you end up having is the amino acid sitting at the back here. And then you end up having your complementary pairs pairing up like that. Now what happens is when they join together, you actually end up leaving behind, let's get rid of that. Oh, that's actually too much, smaller one. you end up leaving behind the amino acid at the back here. And then another tRNA comes in. So then a different one that is carrying the complementary bases here, the complementary anticodon, which, by the way, I didn't label here. This is the anticodon. The anticodon matches with the codon, and it delivers another amino acid. And so I add on another amino acid. And then what happens? Another one appears, and we join the third and final one in my list here. And what you end up getting is this chain. And so what happens is when they drop off the amino acid, the tRNA will go off, but now do you see at the back, it's lost its amino acid because it's dropped it off. So it comes in, drops it off, leaves, and it will go to collect another one that corresponds with its anticodon. We leave behind these amino acids. And the bond that they have in the middle here is called a peptide bond. Not, I repeat, not a polypeptide bond. Please, 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 never, ever, ever call it a polypeptide bond. Okay, it is a peptide bond, that bond in between the amino acids. What you're trying to make, and I think the reason why people make this mistake, is you are just not the same thing. Polypeptide is 50 or more amino acids together. So in order to get that, you need 50 or more amino acids to get a polypeptide. Not the same thing, everybody, as a peptide bond. The bond is the thing that is holding the amino acids together. When we talk about a protein, like the product, we call it a polypeptide, and that's 50 or more, okay? And that's pretty much protein synthesis in a nutshell. But now, what about mutations? Okay, so essentially mutations can occur in two places. They can occur here, where there is incorrect transcription happening, and generally that's the most common place that it happens, and that's where they're probably going to ask you, something incorrect has been covered here. There can also be an error here in translation, but I don't foresee them ever asking that because it's a bit more advanced and beyond high school level. So we're just going to do the transcription level first. So basically what happens in exams is they will say something like, the original code is the following: CCAUGU, let's think of just UAAA. That's the original code, okay? And then they tell you that the following mutation has occurred. Right, so they give you the two codes. Now you have to identify where the code or where the error is. So remember that we write our code in threes. So you got to read the letters 3 at a time. So the first 3 correspond. But if you read the next 3, you will see these do not correspond with one another. There's been an error. And you will notice that we have changed 2 of the nitrogenous bases. So it's, instead of a U, it's an A now. Instead of a G, it's a C now. The U is still the same. Now, when answering this in an exam, in the exam, you must always tell me what it was and then what it has become, because that often gets you a mark. The next thing that you need to do is you need to check on your codon table. Now, often they give you, that little codon table on the side there, and they'll go like, oh, this is the codon, and then this is the amino acid, right? They give that to you on the side. And then they'll say something like the code for UGU, and then ACU, and then let's just do another one like AAA, okay? And then they've got like proline, let's go leucine, and then, what's another one? Alanine, right? They give you that. Now, what you need to do is you need to double check that the codons have changed, but did they actually change the amino acid? Now, in this instance, they have. And now you must acknowledge that. You have to say in your answer that originally it coded for proline because UGU was the original. So it was proline. It's now coding ACU for leucine. So you get a mark for saying that. But you must always double check that, everybody, because sometimes that's not the case. Sometimes you must double check your work and you must double check the table. Sometimes two different combinations of codons code for the same amino acid, which is possible. If you've seen the very big codon table, the one that has all the amino acids on it, you will see that there are many codons that code for the same thing. So just double check that they don't both code for proline. If they do, you still acknowledge that. Now, here's the difference. Let me explain. If it is proline and leucine, because there's a change, you're going to say that there's the change. There it is. And now you're going to acknowledge that there is a different amino acid. And then you're going to say, therefore, potentially a different protein. You could also substitute the word in incorrect amino acid, incorrect protein. Same thing. What happens if it's the same? So what happens if it's proline? Oh, and it's still proline. Even though the codons are different, the amino acids are the same. Well, you change it at the bottom here, and you would say it's the same amino acid, therefore the same protein is formed. So just be very careful and always refer to your little diagram, your little, what do you call this thing, table, just to make sure that it actually is a different amino acid and it's not the same one. And then the last thing they might ask you here is, what's the type of mutation here? The type of mutation that you see here is a gene mutation. This is called a gene mutation. You don't need to know the specific names. I know some of your teachers might teach you point mutations, deletions, insertions, and blah, blah, blah. You don't need to know those, everyone. You need to know just the main type, which is a gene mutation. The other kind of mutation you get is a chromosomal, and that's a whole extra chromosome, whereas this one's just when you change one simple gene. Okay, I'm going to have a look at the chat quickly. If there's anything that I need to clarify on this particular section before I do that little warm-up activity on human evolution. I do also think that people are like chatting heavily in the comments, which is fine, but I'm just hoping that you're also listening and you're not too focused on what people are saying, because then I think you're missing the point of the live lesson as well. Okay, Michaela asks, is there any chance they could ask about a polypeptide bond? No, Michaela, because it's not a thing. It doesn't exist. As I said before, a polypeptide bond doesn't exist. There's no such thing. It's a word that students have made up by putting two concepts together, and it doesn't exist, so they'll never ask it. Okay. Let's have a look here. I'm just having a quick read. See here. Okay. Okay, somebody asked here, why when mutation occurs, protein chain does not change? Okay, so Spongile, I think I've answered your question here, but I'm just going to make sure that everyone heard me say it. You can have different codons, but they end up coding for the same amino acid, even if a mutation occurred. So even though the letters changed, you must double check that they actually code for a different amino acid. If they don't, and they both code like this one I rewrote here, they are different codons, but they code for the same amino acid. The mutation will have no effect on the protein that is made. It'll be exactly the same protein that is made. Okay. Let's see. What else? Let me see if there's any other thing. The Poetic asks, ma'am, is a stick diagram a nucleotide? Yes. So, you know, in your exam guideline, they say you need to be able to draw or label a stick diagram. This is a stick diagram. This is it. This is it. This is what they mean by stick. Okay. They mean not like 3D. Okay. I do see the request for cloning and mutation. We will get around to that. Don't you worry. Mendel, we'll get around to that. Ma'am, does the codon or the anticodon code for the amino acid? It is, oh, it's a good question. I get that one actually often, and I wonder why there's a confusion. But the codon codes for the amino acid, not the tRNA. So the codon codes for the amino acid, not the anticodon. Okay. Oh, DNA calculations. Let's quickly do that one because wowzers trousers, guys. I saw one of these last year and many of us couldn't do this one calculation. So I don't know if you remember this one from last year or you've tried this one, but I'm going to have to sort of go off memory. But the question was something like there is 19 nucleotides in a DNA strand, okay? And they basically had like a table, and I'm just going to really, everyone, I'm going to try and do this from memory only. Oh, let's make this better. No, actually, I'm going to start again. This is irritating me. That doesn't look like a table. Okay, so we had a little table here. And in our table, it's got our, there we go. It's got A, T, G, C, okay? And then they told us we've got 19. Now I need to do quick math here quickly. Let me get my calculator as well, which hopefully everybody will have their calculator with them tomorrow as well, because you always need a calculator. So they said you've got 19 nucleotides in a strand, and then this was strand 1, and this was strand 2. And then they said something like there was 9 in 1, and then there was 4 and 3. Okay. So that's all. Is that all the information they gave us? No, they didn't do 4 and 3. I believe they went 4 and they went 3. Okay. So what you had to do now was you had to figure out how many thymine are in strand 1. So let's not make the mistake that many other people did in this. So this is the first mistake people did. They basically looked at this and went, well, if there's 9 A adenines, there must be 9 thymines. The answer is no. Immediately, no. Okay? I know what you're doing with your eyes, and you're thinking that that must be the case, but that's not true at all. You actually need to figure out what the other letters are first before you can calculate what T is. So let me explain. If there are 19 nucleotides in a DNA strand, that means that the total of the one strand here must equal to 19. So we've got 9 already here. We've got 4 here. We don't know what cytosine is, but we've been asked what thymine is. So now we need to figure out what cytosine is. Now, here's the thing. They have told us that there are 3, no, there should be 4. Let me just make sure, let me do my math quickly, because I'm doing this off memory, guys, and it's frustrating me if I don't get this right now. They want to know, so then it should be, I'm going to quickly do the maths in my head before I say it out loud. If there's 4, then there should also be 4 cytosines. Yes. Oh, I see what I've done here. Because it must go sideways. I'm sorry that I'm going backwards and forward on this one, but I need to get it right. Otherwise, I don't want to teach you something where it doesn't make sense. So if there's 4, 1, 2, 3, yeah, okay, there we go. It's like that. So we now need to figure out what's missing. So what we need to do now is we need to calculate what the guanine is. Now, if there's 3 cytosines in strand 2, there must be 3 guanines. Let me do it in a different color. There must be 3 guanines because these 2 are opposite one another, right? They're complementary. If cytosine is 4 on the one strand, then it needs to be 4 on the other side. Do you get what I'm saying? Because these are complementary to each other, like that. Now we go over to adenine and thymine. Now, if there are 9 adenines on this strand, it means that in the opposite strand, there must be 9 thymines. But now what's left over? Because whatever's left over will tell us how many thymines are in one, and therefore how many adenines are in strand 2. So if we add them all together, and if I put them in my calculator, which I'm not sure if you can see my calculator, but we go 9 plus 3 plus 4, that gives us 16. And we know that there's a total of 19, which means if we say 19 minus 16, we'll have 3, which means that the answer we are looking for is 3 thymines. And then that would also mean there are 3 adenines. And I think the reason why this confuses everybody is they think they have to read the table like top to bottom. So if it's 9 here, it must be 9 here, and 3 here, and it must be 3 here, and so on and so forth. And that's not the case, everybody. You've got to read it diagonally. This is from a past paper. I think it was last year's past paper. Or not last year. Was it last year? No, 2022 final exam. That was this question. It was in the multiple choice section. And then when you're doing like percentages, like if they tell you like 40% is thymine, how much is cytosine? Do you know how to work that out, everyone? Do you want me to go over it again? I'm looking at the chat and it's kind of frozen. Do you want me to go over this again? People are saying yes. Quite a number of you are saying yes. No's and yeses. Okay, I'll do it very quickly and then I'll do the percentage. Don't worry, I can go fast now because I figured out. The math is mathin', babes. The math is mathin'. So this was 9, right? Okay, let me rub out everything here so we can start fresh. I'm also going to give you a visual to work with this time because some of us are more like, we like to see it being done versus me saying it verbally. Okay. So I'm going to move this out of the way. We'll come back to that question shortly. Okay. How did I fill this in? So we're looking for thymine, how many thymines there are. Okay. So if in strand 1, if this is strand 1, and you can literally draw this out if you really want to, guys. There are 9 adenines. So we go 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 adenines in strand 1. That means in strand 2, because it's complementary, right? There should be 9 thymines. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay? So that means 9 adenines, 9 thymines. If we go to our table, 9 adenines, and we go diagonal, because remember it's how many thymines? There are 9. So it's diagonal. Do you see that? That's not how many thymines there are in 1. We still don't know that, but we can come back to that because we need to calculate these others. We are told that in strand 1 there are 4 cytosines. So let's do that. 4. 1, 2, 3, 4. On strand 2, there must be 4 guanines because they're complementary. So 4 on this side, 4 on this side. Yes? So we go up. 4 cytosines means 4 guanines. Now, it says there are 3 cytosines in strand 2. How many guanines? Well, if we go down here, and I literally write that in, and I go 1, 2, 3. There are 3 cytosines. On the opposite side, there should be 1, 2, 3 guanines. 3 and 3. So we put that in the table. Let's do it in green so we stay on track here. 3. Do you see that was diagonal? So you can actually figure them out in a diagonal pattern. Now that we have figured out 9 is there and 3 and 4, we fill that in. We still need to figure out what thymine is in strand 1. Now you see you can't read a diagonal here. But what you can do is you can add together 9, 3, and 4. And that gives you 16. When you subtract 16 from 19, it will give you 3. And there are 3 thymines in strand 1, and that was the answer that we were looking for. Okay, I think I've answered everyone's question now. And so I'm going to go back to what I was asking earlier, and I think I've clarified that very straightforward. And if you want to do this drawing out now where I just literally drew out the whole strand, you have time, guys. You do have enough time to do that. Okay, so the question I asked you was, if you had 40% thymine, how much cytosine do you have in a molecule? So remember that this works in pairs. So that means if we have in a DNA molecule 40% thymine, it means we must have the equal amount of adenine on the other side. So we also have 40%. But here's the thing. DNA is made up of 100%. So that's like the total amount that you have. We've already allocated 40 and 40, which gives us 80%. So that means 80% of this DNA molecule is A's and T's. That means the difference between these two is 20%. That 20% must evenly be shared between cytosine and guanine, because they must always be complementary. And that means that you get 10% cytosine and 10% guanine, because it's all about calculating the total that you have. And so remember, this is the total that you have. You were told you had 40%, which means you must have 40% adenine on the other side. Our DNA molecule is always out of 100, so 40 plus 40 gives us 80%. We subtract that from the 100, which gives us 20%. That 20% must be divided, very importantly, equally between the remaining bases. So that 20 halved gives us what cytosine is and what guanine is. Okay, right. I'm going to move on to this human evolution question. I think that that's what all of us also want to see. And I want to do a meiosis question with you as well. I think that's really important. Okay, right. So It says the diagram below shows the evolutionary relationship between different species. And so you should expect to see something like this tomorrow. If it's not something exactly like this, it'll be something very, very similar. It might even have their skulls on it. So just take some time to note things like where we are. So there's us over there. And then you'll also notice that we've got some other organisms here that are not us. So like gorillas and troglodytes. But we can locate us easily. And then we've got A, B, and C, which probably are going to be things that we need to identify or list or label. So let's go up to the first question. It says, identify the type of diagram above. And everybody, I'm hoping that we all know that this is a phylogenetic tree. Okay, not a phylogenetic tree, a phylogenetic tree. Then it says give, for our next question, it says give the genus that gave rise to Australopithecus at A. Right, so we go up here. Now, what is a genus, everybody? Let's remember and let's remind ourselves. We have got, when we give things names, we've got the genus, and then we've got the species, right? So the genus names comes first, and then the species name comes next. Everyone is asking me if they could also say cladogram. I think they probably would accept that too in the exam, yes. I would go with phylogenetic tree, but I think they would make an allowance for this to be a cladogram as well. So, what does give rise mean? So, that's a great question. I just saw that in the chat now, this bit here, give rise. It basically means where did it originate from? So, that's what that means. Give rise means originate. So, give the genus that originated before Australopithecus. So if we go to our diagram, here is Australopithecus. Does anybody know the genus before Australopithecus that we need to know? Let's see if anyone puts it in the chat. I'm just seeing. Yes, it's Ardipithecus, everybody. This is Ardipithecus, or Ardi, okay, for short. It's Ardipithecus. So Ardipithecus is the genus where Australopithecus arose from. Okay. Then it says, give the letter, let's highlight that, the letter that represents the most recent common ancestor for Homo sapiens and Homo erectus. So let's go up to our picture here. We've got Homo sapiens and we've got Homo erectus. So the letter of their most recent common ancestor, guys, that can only be C. That's the last time they both shared a common ancestor, because you see how it branches off to the left here and then to the right? That means that they went on separate journeys and therefore became separate ideas or separate groups. So I'm going to put the letter C there. It's a nice easy one. Then for the next question, it says, name two species that existed at the same time as Homo erectus. So we're going to go, and specifically we're looking for species, hey? So be careful of what names you give. So we're going to look here, and it says, what was it saying? Erectus, okay. So at the same time, now if erectus is around at this time, at the same time, right? What you need to do, and I have actually seen the memo for this, so I know what the answer is. If you're looking at this diagram and you're like, ma'am, I don't know what to say, what you need to do is you need to look left and right to see where it intersects. So if I took a ruler like that, okay, and I look at which organisms were around at the same time, there are only two that it intersects with that has not deviated again. So what I mean is, the answers we are looking for is the troglodyte and the gorilla. Now, before you ask me, why not Homo sapien and Neanderthalensis? Because do you see they split here? That little split, if we go close, this little split here basically means that they, oh, this is going to irritate me now. Let me go out. Sorry, I've moved my page. That little split over here means that they haven't formed yet. So there is no interaction there whatsoever. So if I draw that line again and we go over and we go, oh, I keep moving my page. Sorry, guys. You see where I have my line here? If I drew it nice and straight, it would be even better. But what you can see here is there is no interception there. I'll even draw it nice and straight. There is no intercept. So do you see that they originated after where the line intersects? So they were not around at the same time as Homo erectus. The only individuals that were around were the gorilla and the troglodyte. So that's going to be our answer for that. It gives the species name. So the species is our, what was it? Gorilla and troglodyte. Or I'm not spelling gorilla correctly. Let me get rid of that. Or what is it? So how many L's? Oh, it's 2 L's. Gorilla and the troglodyte. Okay. Then it says for number 4, give one example of the fossil Australopithecus africanus that was found in South Africa. You should automatically know this, but I'm hoping that we know it off the top of our head. Does everybody know one fossil of Australopithecus africanus that was found in South Africa? Somebody asked, do we underline the name of the species? Yes, we do. Like that, because that's a species name. Oh, so many names. I'm wondering. Does everybody, I'm worried that not everybody knows a fossil of Australopithecus africanus. It worries me only because it is, it's something that they will ask you. Let's see. Are you all giving the right answer? Somebody said, ooh, no. I'm looking at all your answers and I'm hoping for the best here, guys. Okay, you've got 2 answers that you could give. We're either going Mrs. Ples or Taung child. That's your other option, guys. So you could give either answer, not Lucy. For those of you who said Lucy, Lucy is afarensis. Afarensis. That's the one before Africanus. So Mrs. Ples or Taung child. Okay. Right. I probably am going to guess there's lots of questions on maybe human evolution. And I do have another little human evolution question that I want to do. But I also wanted to cover some meiosis with you. You might have seen my TikTok yesterday where I said this is probably the question that's going to catch you, that you don't think it's going to catch you. You don't think meiosis is going to get you. But it's these kinds of questions I'm going to show you now that often you lose 6 marks because you don't know what to say and you make an error. And because we didn't draw anything on Monday's exam, I'm thinking we're probably going to have to draw something tomorrow. And in all likelihood, it's going to be a graph or it's going to be something you have to draw in meiosis. Okay. Oh, and if everyone is asking about Littlefoot, yes, it could also be Littlefoot. That was our third option as well. You could have said Littlefoot, Mrs. Players, or the Town Child. Those are your three options. Okay, so let's do this meiosis question because I think it's quite important to touch on, because I think a lot of you are probably not spending that much time studying meiosis and you're spending way more time studying other stuff. So it says the diagram below represents metaphase 1 of meiosis. So be grateful that they're even telling you what it is, because a lot of the times they don't. They just expect you to know that this is meiosis, and they need you to know it's meiosis 1. Before we go further, how did I know that this was meiosis 1 without even being told? Number one, the first thing is the chromosomes have aligned at the equator. So that's how I know it's metaphase. But how do I know it's 1? I know it's 1 because our homologous pairs are still visible, like they're next to each other. If this was metaphase 2, you would just have one chromosome lying at the equator. And yes, it would have its little like colored in pieces as well to look like that, but it wouldn't be 2. So when you see 2 at the equator, you know we're in meiosis 1. If you see just 1 at the equator with its little spindle fibers attached to it like that, we now know we're in meiosis 2. So that's the important thing I just wanted to point out to you. And let's quickly do the labels for A and B. So A is pointing to our little structures that grow our spindle fibers, and I'm hoping we all know that that is a centriole. I'm hoping we all know that. Just double checking. And then B is often misidentified as the nuclear membrane, but it's not the nuclear membrane because the nuclear membrane has dissolved. It's not in this picture anymore. This B is the cell membrane. The nuclear membrane would have been around here, like in this dotted line all the way around. But it's not there anymore. Remember, it disappears, it disintegrates right at the beginning in prophase. So there is no more nuclear membrane, it's a cell membrane. A quick question before I continue. Megan asks, how do I know the difference between a centrosome and a centriole? Lovely question. I'm going to tell it to you very quickly. So the centrosome is a round temporary organelle that has the centrioles inside of it. So this is the centriole. This is the centrosome. And that thing only ever appears when meiosis hasn't even begun, like when we're in interphase, because remember there's the nuclear membrane, there's the chromatin network. This is the centrosome with the little centrioles inside of it, and then that's the nuclear membrane with the chromatin network. So you only ever see the centrosome when a cell is not dividing, when it's in interphase. I hope that answers your question, and so that we're not too confused about the difference between those two things. Okay, so now that we've done that, let's answer our question. So identify part B, which we already have. It's the cell membrane. Then it says state one function of part A. So remember, part A is the centrioles, and what is one of their functions? I would write here, it grows spindle fibers. Okay, easy one. Next one. It says explain the contribution of metaphase 1 to natural selection for 6 marks. Okay, so now I want you to take a moment here and I want you to think about what you would say for 6 marks. And this is why I made the TikTok I did yesterday saying that this is where you could potentially not be prepared enough and know what to say. It's a big question, everybody. And because it's an explain question, you have to be very, very thorough in your explanation. Now, remember, as you think about what you want to say, explain questions must always have causes and effects. Now, this is out of 6. which means you're going to need at least one cause and 5 effects. But I actually think you need, for this one, you actually need 2 causes and 4 effects. In other words, one cause will have 2 effects, and another cause will have another 2 effects. So you're going to have to structure this really well to get 6 out of 6. Okay? And I'm going to show you it's not as scary as it sounds. You think you don't know what to say, but you do. So I'm going to break it down for you. This is what we're going to say. Okay? First of all, let's just remember something. We've got natural selection that we have to talk about, and we've got metaphase 1 that we have to talk about in our answer. We have to reference these 2 things together. You know enough. I promise you do. You know enough to get 6 out of 6. So remember, natural selection. What is it all about? If you remember the description of natural selection, it's all about variation. Okay? It's even in the opening line of the natural selection description. It's in the opening line, everyone. To prove my point, I'll show you where it is in the opening line because I have a copy of my cheat sheet study guide. Let's see, is it in this one? Or actually, I have, let me just get my study guideline. I'm sure I have it with me. Let's see. Do I have my guideline with me? Oh, I thought I had my guideline. Let's see. No, I don't have my guideline with me. I thought I had my guideline with me at home today, but I don't. But the very first thing is variation. Now, that's the goal, variation, right? Metaphase 1, what occurs at metaphase 1? We have random arrangement that occurs. Now, how does random arrangement contribute to variation? You now need to talk about how the chromosomes randomly arrange at the equator, which means that there will be a variety of chromosomes that are going to lead to an increased level of variation. So how do we explain this for 6 out of 6? Somebody in the chat, I'm just reading, and that's why I pause sometimes because I'm reading what everyone says. It's not crossing over, everybody, because it says, explain the contribution. Let me show you the question again. Explain the contribution metaphase 1 has to natural selection. So let's remember what random arrangement is. Random arrangement is when our paternal and maternal chromosomes align at equator in a random way. So if you don't know what that means, I'm just going to draw you a little sketch. What you have in every couple or every pair of chromosomes is a paternal and a maternal chromosome. Now, the way in which they line up at the equator like this is random. So green and pink or purple. Okay, this is the paternal, this is the maternal chromosome. The way they arrange like this is random. So we've got 2 maternals this side and 1 paternal. And on this side, we've got 1 maternal and 2 paternals on the other side. So when this divides into 2 separate cells, This cell is going to have more maternal information, and this cell is going to have more paternal information. And so that increases the chances of variation, or it increases the variety, and it makes these two cells that they're going to form very, very different. Okay. So someone is asking before I continue any further, because I can see a lot of questions about this. Ma'am, does this fall under the concept of Gregor's law of independent assortment? This is not independent assortment, everybody. Well, let me explain. Independent assortment is not the same as random arrangement. Obviously, their names are different, therefore their outcomes are different. So random arrangement means they randomly arrange at the equator like this. Independent assortment, what that means, is when you inherit alleles independently of one another. If you want the simplest explanation, that means like your blood group is inherited separately from your hair color. They're not inherited together in that way. So you don't get your hair color and your blood group together. They're independent of each other. That's what independent assortment means. It's not the same thing. Okay? So now that we have a bit of a background of what we're going to talk about for 6 marks, now we're going to structure this really well, because that's how you get 6 out of 6. So let's go cause. So remember, if I just quickly go back to the question, because I want to refresh my own brain and my memory, explain the contribution of metaphase 1 to natural selection. So the contribution is the following. So the cause. Metaphase 1, I'm going to write it out in full. Metaphase 1, I'm going to say introduces. Metaphase 1 uses random arrangement of the chromosomes. Okay, now we go for all of the effects. So let's go effect. That random arrangement of chromosomes means that there is a random inheritance of paternal and maternal chromosomes. So there's a random inheritance of maternal and paternal chromosomes. Next point. That random ratio that you have will, this is my next effect, produce different gametes. Next point, which increases genetic variation. Next point, when you increase genetic variation, you increase the, I'm going too fast, the chances of survival. Okay, I'm going to pause there in case I'm losing anybody, and I'm just going to explain from the top. So I've given my cause. My main cause, if I just highlight it, is random arrangement. So the cause here is metaphase. Metaphase 1 uses random arrangement of chromosomes. That's the cause. Now, these are all the effects, okay? The random arrangement leads to a random inheritance of maternal and paternal. So I'm telling them what that is. That then produces very different gametes. Those very different gametes means that we have an increase in genetic variation. If you have an increase in genetic variation, you also increase the chances of survival. Now, that means at this stage we have, if I were marking this, we've got 1, 2, 3, 4, 5 things. We just need our final thing, which if we go back to the question, is in reference to natural selection. So if we look back at my last point, increases chances of survival, we can finally say it assists in natural selection by improving the variation of offspring. Now, I know that the last few points sound similar, but they're not. And I'm going to tell you why they're not. The key differences is that this point is just talking about genetic variation in general. This point is talking about chances of surviving. And this point here is talking about the actual babies and offspring. So I know the last three feel very similar, but they're not all the same thing. They are speaking to slightly different things. Okay. Is everybody okay with this one? It's actually not that bad. If you follow the structure like this, if you go cause and effect with your effects, it's not too crazy. I think the reason why we become overwhelmed with these 6-mark questions is we feel like we don't know enough to say 6 things. But what you have to do is you have to do the following. Just think of this tomorrow in your head. I know what random arrangement is, so I write it. Then I must tell the marker what it is. So I say random arrangement is the inheritance of the maternal and paternal chromosomes randomly, right? So you tell them what it is. Then you tell them the products, which is the fact that you'll make different gametes. And because you make different gametes, you'll have a variety or an increased genetic variation. And when you have an increased genetic variation, what's the product of that? Increased chances of survival. And when you have an increased chance of survival, you have a variation of offspring. So you have a variety of offspring that could be living. So it's like, do you see how one thought falls into the next? It's like, therefore, therefore, therefore. That's maybe another way you can think of it tomorrow. You make a statement, so the cause, and you go, it's the inheritance of paternal chromosomes, therefore there's different gametes, therefore it increases variation, therefore it increases chances of survival, therefore it improves the variation within the offspring. And that's all linked to natural selection. And they do like asking this question, everybody. A lot of people in the chat are talking about crossing over. I just want to point something out to you. Crossing over doesn't occur in metaphase 1. So you can't talk about crossing over. And I think that's where a lot of you maybe are going to make a mistake tomorrow. They're only talking about metaphase, please. There is no crossing over occurring in metaphase. It happens in prophase. Okay. Baby Lou says, will meiosis always be connected to natural selection? Because can't we then prepare for each question like an essay? Yes, you can actually, because meiosis can be linked to natural selection and speciation. So it can be connected to both. It can also, let's not forget, by the way, it can also be connected to genetics, like genetic abnormalities, like people having too many or too few chromosomes, which is actually what I want to get to today. In case we do a drawing tomorrow, let's practice this one here. So this one here, our next question says, the diagram below shows 4 possible daughter cells formed at the end of meiosis. Here they all are. It says, for 234, draw cell D, including the missing 4th chromosome. Now, you need to pay careful attention to how they've colored in the chromosomes. So we have to redraw this one. And so I'm going to draw it at the bottom here. And so when you draw this tomorrow, if you have to draw anything, you must draw everything as you see it. That means the exact shape and the exact color. So we're going to redraw this exactly as they have. Do you see how they've made it crooked like that? You are also going to draw it at an angle. You are not going to make them straight. You are not going to change anything about them. You are going to draw it just as they have done it, exactly how they've colored it in, and the same size. Okay, so just a little checklist. It must be the same size, it must be the same shape, and it must have the same shading. Please, please, please. Those are the things tomorrow if you have to draw this. Please make sure you get it right. Then we've got this little chromosome. Looks like the letter R. And then we've got this one that looks like a bit like the letter Z. So draw it exactly how they have drawn it. Okay, which I've done now. And I'm going to put my membrane around it. Now you have to draw the missing chromosome. So how do I find the missing chromosome? I go back to the original picture, and I'm just going to clean it up a bit so we can see it nice and clearly. So now I'm looking for a chromosome that has this composition at the bottom here. So I need a full black chromosome or full shaded in chromosome, and I need 2 white chromosomes. And if I look at all the others, these are all partially colored in, which means that these 2 were probably from the same group, and then these 2 are from the same group. But I want you to know and notice that we've got 1 white one and 3 shaded black chromosomes here. And then we've only got 1 black and 2 white chromosomes here, or unshaded, so shaded and unshaded. Now, if you know anything about genetics, you should know that if there are 3 shaded and 1 white, there's probably going to be 3 unshaded and 1 black or 1 shaded chromosome here. And to prove that, if we go up to the top, let's look for the 3 shaded, 1 white. Let's see which side of these chromosomes has that. So there's a black one, a white one, a black, and a black, so shaded. So let me highlight it for you. Yeah, let's stay with pink. So 1, 2, 3, 4. Those 4, do you see? Solid black, a solid black, or a solid shaded, and 1 unshaded. Those 4 went into this this cell here. These are the mixed and matched ones, which if we go up here, you can see there is the mixing and matching ones, which means that we now need the other side. And the other side is this white one or unshaded, this shaded, unshaded, unshaded. And you can even see their shapes are the same. So the missing chromosome from here must be in our cell. And right now we have an unshaded, and a shaded. We now need a unshaded one. So if we go down here, we're missing 1 unshaded one. But here's the important part. You have to get the size, shape, and shading right. So we know that it's unshaded, but what is its shape and its size? So we've got this one that looks like a Z and one that looks like a little letter R. Let's look for the missing one. So we've got one that looks like the R over there, one that looks like the little Z. There is the shaded one. So this last one, you see, is straight. You have to draw it straight, like that. If you don't get the shape right and the size, you are not going to get your marks tomorrow. Okay, and if you had to label this, I'm hoping that you don't have to. All you would have to do for the 3 marks is you would have to also give this a heading as well. And our heading could be something like, let me start off with the correct way. This is a diagram showing, I can't fit it in all now, but daughter cell D. Okay, and that's what you're going to draw. You don't need to provide necessarily any labels. You can if you want to, for safety's sake. This would be the cell membrane. This would be the nuclear membrane, or you could even go nucleus. And then if you want, this is the nucleolus, by the way, this little round thing. And then these are chromosomes. Oh, I can't fit it in. I've run out of page. Chromosomes. and you can leave it like that. But it's for 3 marks. So the likelihood is that you're not actually getting any marks for labels this time around. What are you getting a mark for? You're getting a mark for the heading, and you are getting a mark for the correct number of chromosomes. So it'd be like N for number, and then D for drawing. Like, did you correctly draw the right one, which is a single straight unshaded chromosome? Yeah, I don't think I saw the comment now. I don't think it's going to be drawing and labeling for 3. I think it's just going to be drawing, but please also always put a heading, because you often get a mark for the heading and then 1 for the correct number of chromosomes and 1 for the correct drawing of chromosomes. Somebody said here, isn't it chromatids? No, these are not chromatids. I'm going to ask the question as a rhetorical question, but how do you know these are not chromatids? Chromatids are only chromatids when there is still a centromere present, like this. Centromere, chromatid, chromatid. The moment they separate like this, they're chromosomes. So these are chromosomes, not chromatids. You could also say single-stranded chromosomes if you wanted. Okay. Alrighty. I think what we're going to do to wrap this up is I'm going to have a look at the comment section. We can do a human slash, I'm just going to show you the top question here, which is a speciation through geographic isolation, and then a human one. Or I can explain some genetics. You got to decide. That's going to be our last session for today. Either the human question here with a little bit of geographic speciation at the top, or we're going to do genetics. And I'll do like I'll walk you through a pedigree diagram or a genetic cross. What do you guys want to do? Oh, it's hard because everybody is. I'm actually going to put a poll. Okay, so let's vote in the poll. Quickly, I'm going to type it up because it's hard to see all of your suggestions or what you want to do. So I'm going to put genetics, or I'm going to go human evolution. I'm going to put it in the chat now, and I want you to vote. I've put the poll up. Vote in the poll, guys. You should be able to see the poll. Somebody says just do the hardest one. That will be human evolution will be the hardest one. Let's see. Right now, human evolution seems to be in the lead. Still human evolution in the lead. I know some of you find evolution very easy. The hard part is both genetics and evolution are the biggest parts of this paper. They're the 2 biggest sections. Okay, it looks like everybody is voting for human evolution. You should be drawing in pencil. Someone's question said pencil or pen. I know people want both. It's quite tricky to do both with the time that we have left. Let's see here. Okay, human evolution has stuck in the lead this whole time, everybody. If there's a poll, I'm going to end the poll now because human evolution's in the lead. Okay, I'm going to go quickly through this first bit. So it says describe the process of speciation through geographic isolation. I'm not going to spend a huge amount of time on that because do you know what you need to do for 7 marks, everybody? This is the easiest. This is what you hope for tomorrow. You hope you get something like this. I don't think you're going to get this because they already asked this in the supplementary. This question is also from the supplementary paper. I think tomorrow they're going to ask an application question of geographic isolation. But if they ask you for 7 marks, what do you write? Well, this is just a little extract from my study guide. This is from the guideline itself. This is how you explain speciation with a geographical barrier. So all you're going to do is you're going to say if a population of a single species becomes separated by a geographical barrier, if they tell you the barrier, you must write the barrier in. They split into 2. There is no gene flow between the 2 populations. And since each population may be exposed to different environmental conditions, if they tell you the condition, like hot, cold, no rain, lots of rain, flooding, whatever it is, you put it in. Natural selection occurs independently. The individuals become very different from another, geno and phenotypically. And if they ever were to mix again, they will not be able to interbreed. And now we have 2 populations that are different species. This, everybody, is a template answer. In other words, this is what you use to apply to the question. If they give you an example of an animal, like my little picture here was of these birds, right? So this bird becomes these 3 species. You have to talk about how this bird became these 3 species through speciation. You talk about the barrier. In this case, it's land and ocean. They've got ocean in between them, which is their geographical barrier. There's no gene flow between these populations. They each have independently experienced their environments. Maybe this island is very hot and dry. This island is covered in fruit trees. This island is mostly insects and cacti and nothing else. And because their environments are all very different, they experience natural selection differently, and they become so different geno and phenotypically that if they mix, meaning if they meet again, they will not be able to interbreed, and they will now be 2 populations, or in this case 4 populations, who are very different species. So you just take this very simple answer, and all these words that I've bolded here, those are your key points. They are the ones that you need to make sure are there. If it's not 2 populations, it might be like in my one up here, 4. You must just match it to however many that you have. Okay? And that's it. 7 marks. Done. Closed. Let's do the actual hominid one now. So this question says the diagram below represents the skulls of hominids. We've got Homo sapiens, we've got gorilla, and Homo naledi. And they're helping you out here by even labeling that this is the cranial ridge. So number 341 says list 3 similarities in relation to vision that are shared by these 3 organisms. I'm very curious to know if you know what they share. They've got 3 things that they share. Number 1, they have color vision, or they have cones and rods, which means they can see in color. Do we know what another one is? The second one, they've got binocular vision. And anything else? Color, binocular vision. Stereoscopic also means binocular, everybody. They are the same thing. And the last one is, I know it seems the same, but the eyes are in the front. These 2 are not the same thing. Binocular vision and eyes in the front are not the same thing. The eyes in the front is the location. The binocular vision is the overlap of their vision. In other words, one eye overlaps on the other. Okay. So that is the 3 similarities in relation to their vision. It's a bit of a tricky one, hey, because I think the first 2 you guys would have been like, boom, I got those. You weren't so sure maybe about the third one. Okay, number 2, name 1 species in the diagram that was most prognathous. And so remember, prognathous refers to the jaw. So we're talking about this structure here. Now, if you look at these 3 skulls, who has the most prognathous jaw? I think this is an easy one, but it's going to be the gorilla. Okay, has the bigger, look at that prognathous jaw there. Okay, let's move on. Still easy peasy stuff at this stage. Number three, it says describe two structures, and so it's a describe question, that cause the species named in 342 to have the most prognathous jaw. Now, this is an interesting one, and I chose this question because it's a little bit harder than some of the other questions we've had in the previous years. And so I'm hoping that we know. So it says describe two structures that cause the species, so gorilla, to be most prognathous. So they've actually given you a clue up here, which is the cranial ridge. That's actually one of the structures you need to talk about. So describe two structures. So describing the two structures would be the cranial ridge and this guy over here. Do you know what that is? Because it's also linked to the prognathous jaw. That is does anyone know what it is? I'll talk about the teeth now, everybody. Don't you worry. But the teeth is not directly correlated to the jawbone. It's the brow bone, everybody. The brow bone and the cranial ridge. It's also known as the sagittal crest. Okay, that's the other word that you can use for it. The brow bone and the cranial ridge are linked to the prognathous jaw. Does everybody know why? I think that's actually a very good question. I don't think everybody knows why they're linked to each other, but I'm going to explain now why those are the answers. They are linked to one another because a prognathous jaw was used for biting and like big, heavy chewing. And when you chew, what do you need besides a big jaw? When you chew, you need really big muscles. And so the muscles that come from the jaw, if I just get rid of this, the muscles that come from the jaw attach onto the cranial ridge and onto the eyebrow bone like that. And so that's what allows the jaw to have this hard biting power. And that is what is linked to our two main structures. Okay, so if I go back down to the question, describe the two structures that cause the species name in 342 to be most prognathous. It's the cranial ridge and the brow bone, but that's just telling me the two structures. I have to describe them because that is where we had the largest muscle attachment for chewing. And that's only for 2 marks, so you don't need to go into like crazy detail. Okay, so describe the two structures that caused the species to be most prognathous. Now, why can't I put teeth, guys? A lot of you have said canines in the chat. There's a reason why canines didn't cause the jaw to be this. The jaw caused the teeth, not the teeth caused the jaw. I think that's what I'm trying to ask you. So I'm asking you, why are we saying teeth? Teeth don't cause a prognathous jaw. The question very specifically says the two structures that caused the species to be most prognathous, and the structures that caused it, that allowed it to do its job, was the cranial ridge and the brow bone. Don't worry, this live is recorded, everybody, so when we're done, it's going to go up and you can watch it again. Okay. I hope that I've made that clear for everybody, and I'm going to move on to our last 2 questions. 344 says, Homo naledi was bipedal for most of its adult life. Explain how the structure of Homo naledi's skull would have assisted in bipedalism for 3 marks. So what are the key words of our answer? We're looking for the skull, and we're looking for assisted in bipedalism. Now, I'm hoping that the first and main thing you are going to say is a more forward foramen magnum. Okay, so that's your key. Now it's explained for 3, which means if we go down here, we're going to need a cause, and we're going to give 2 effects, right? So we get 3 out of 3. Cause. So they have a more forward foramen magnum. Okay, so that's going to be our cause. Then you need to tell me about that foramen magnum, like what's its effect? How would have that assisted in bipedalism? That would have assisted bipedalism in doing what? So more forward foramen magnum, how does that assist, everybody? Lovely. I see someone's put there in the chat, said that the more forward foramen magnum is in a more forward position. Therefore, we have a vertical entrance of the spine. And the vertical entrance of the spine creates an upright posture. You could also speak about like the S-shaped spine if you wanted to here. So you could say the more forward foramen magnum means there's a vertical entrance of the spine into the skull, which creates an upright posture. And you can finish that sentence by saying creates an upright posture used for bipedalism. Somebody asked if spelling counts in the exam. It only counts if a word sounds similar to something else. Okay. I feel like everybody wants me to do cloning and they're like shouting it in the chat. Please, ma'am, please. Somebody asked about, I think it's Patience says, what about the pelvic girdle? The pelvic girdle is not directly linked to the skull. It's like a secondary thing. So you would only talk about the pelvis if they were asking about bipedalism in general. Like if they said, what structures assist with bipedalism? Then you can say the foramen magnum, you can say the S-shaped spine, and you can say the pelvis, the short and broad pelvis. Those are the things that assist in bipedalism. But this is just specifically talking about the skull only. So we can't actually talk about the pelvis. We can just say that the foramen magnum is more forward, which allows for a vertical entrance of the spine, creates an upright posture. You could also say creates an even distribution of weight for an upright posture or vertical posture. Okay. All right. I think what I'm going to do, because I want to move on because I want to save time, is I'm going to go on to the next one. Describe the difference between Homo sapiens and gorilla in relation to the shape of the spine. So I'm just going to break it down for us here. If we were to describe the difference, Homo sapiens have an S shape, whereas gorillas have a C shape. Okay. And then pelvis, Homo sapiens have a short and broad, whereas gorillas have a long and narrow. I'm hoping everybody knows that. That's a nice easy one to get 4 out of 4 tomorrow. Wide and short, and or you could say broad also means wide, by the way. So you could say short and wide, or short and broad, and long and narrow. Okay, last but not least, for number 6, it says explain why the gorilla species has a cranial ridge. It actually links a little bit earlier on to us talking about the two structures that cause the species to have a prognathous jaw. It links to, if you're going to explain why it has a cranial ridge, in order to, I'm just going to write here, support large chewing muscles to eat, I'm going to call it fibrous vegetation. So to support large chewing muscles, that's my cause, and my effect is so that they can eat fibrous vegetation. That's why they have a large cranial ridge. Okay, right. Because everyone's begging for cloning, I'm going to do cloning, and that's going to be our last thing, okay? I actually did cloning the other day, and if you want, you can also go look at the lesson on this in my playlist. So this is the last thing we're going to cover today because we've had a long lesson. So here is cloning. This is called reproductive cloning. Okay. Reproductive cloning is when you want to make a copy of a whole animal. So what you do is you take two animals. And in this case, I'm going to keep it simple. Let's say we have pig A and we have pig B. Okay? Pig A and pig B. So from pig A, we are going to take a somatic cell. And from pig B, we are going to take an egg cell. Now, I want you to remember that somatic cells are diploid and egg cells are haploid. So when we do reproductive cloning, we want certain parts of these cells. In the somatic cell, we want just the nucleus. So we take the nucleus out. In pig B, I just want the empty egg cell. I take out the nucleus, so there's the nucleus there, and I get rid of it. I don't want it. I then take my nucleus, my haploid nucleus, so I'm just going to write here, this is the nucleus. Remember, it's not haploid, diploid, I beg your pardon, it's diploid. And I'm going to put these two things together. And I'm going to use a little electrical shock. And I'm going to shock this cell, and this cell, which was the empty ovum, it's also known as a gutted cell, plus the nucleus from the somatic cell, creates, with a little electric shock, a zygote. So it like fools the cell into thinking, the egg cell, into thinking it's being fertilized when it actually hasn't. You've just taken this nucleus and you've inserted it in here. That creates a zygote. And generally speaking, we put the zygote back into pig B because pig B was the original donor of the egg. And so let me just give them some color differences. So pig B is pink and pig A is green. So the nucleus is pink. And the nucleus is green and the cell is pink. So I just want you to see them inside one another so you can see where each thing came from, like that. And so we put the zygote back into pig B because pig B gave us the original egg. And now in an exam, they might ask you, which pig are we actually cloning? We are cloning pig A, because the somatic cell has all the chromosomes that we want and need. Maybe this pig grows big and healthy, and if we want to grow it for meat, we want big pigs with lots of meat and muscle. So we grow a somatic cell, and we can take it from anywhere. We can take it from their muscle, we can take it from their skin. We take the nucleus out of it, and we take our gutted, our empty egg. So we take out the nucleus, we remove it, and we put the diploid nucleus into the gutted cell. So it looks like this. We give it a little electrical shock, and it thinks it's fertilized, and it becomes a zygote. That zygote we put inside of pig B. Why pig B? Because pig B was the egg donor originally, and then pig B will give birth to the clone of pig A. And that is reproductive cloning, everybody. You can also put it into a third pig. So you could put it into pig B, or there could be pig C. You don't have to, but you can. Different explanations have different outcomes, either pig B or pig C. Okay. Now, to kind of wrap up today, I want to tell you a couple of top tips that I think you should prepare for. Number one, you should go and learn Out of Africa off by heart from your guideline. That means that you need to go and learn like where the fossils were made and where they weren't found. And I want you to focus on the words only, and I want you to focus on the words oldest and newest. You'll see those words in the Out of Africa hypothesis, and you have to use them. So you must say, this fossil was only found in Africa. This fossil is the oldest in Africa, but the newest fossils were found elsewhere. You also need to know where they were found. So Ardipithecus is one of them that you need to talk about, and Australopithecus is the other, and Homo sapiens is the third. And so if you learn the paragraph in your exam guideline, that's all you need to learn. But remembering, you must use these words tomorrow if they ask you to explain Out of Africa. The other thing that I want to remind you of, which is easy marks, is please remember your genetic cross layout. So remember that's like P1, and then you go like big B, small b, little b, and then below that, and you also need to write like, let's say this is a black cat, and then this is a white cat. Don't forget to go meiosis, and then in the column here, gametes. And I teach my kids to put their gametes in circles because they never draw them far away from each other. Then I write fertilization, and then I go F1 in the margin, and then I do my Punnett square, and then I do all the cross. So please just remember this for tomorrow, the layout. The layout is really important. So these words and how they're laid out, that's really important. Let's not forget about that tomorrow. And I also want to remind all of you, as I kind of wrap this up, that I do have a playlist on my YouTube where I've done past paper questions, where you can go through them and you can watch me do them. And they're ranging from easy, medium, hard, and there are 1 or 2 advanced questions there as well, everybody. And I think I'm going to wrap up the lesson now because we've been here for quite some time. I'm going to end the live, and then what happens is this then gets saved onto a playlist. So it takes a couple of minutes before it goes live again for you to watch. But you can go back at any point and you can rewatch the video today. And then you can have a look at maybe something that we covered that you want to hear again. I want to say good luck for tomorrow. You're going to do fine. You're going to be fine. You just need to lock down your explanations. That's your weakest point for tomorrow. It's not labeling and drawing and identifying. It is going to be your explanations that are your weakest points. Because unlike paper 1, which is all anatomy, paper 2 is about explanations, explaining speciation, genetics inheritance, variation, explaining meiosis, crossing over, random arrangement, explaining the laws of Mendel. And so you've got to spend the rest of today, everybody, working on explaining those things. Okay, and I hope that you have a wonderful day. Please also get some rest, and make sure, my last top tip is that you attempt this past paper, the one that we've been working on today. This one, the one that we've been doing this whole time, where I got all these questions from. I suggest that you go and you practice the May-June 2024 supplementary paper. I told you can also get it on testpapers.co.za, and you can also get it on the I'll write it again, WCED website under the matric resources, and I actually think they have a memo there as well. Okay, thank you everybody, and good luck for your exam tomorrow, and I'll see you all again soon. Bye.